Let's do some important topics of chapter 9 of class 10th, and this name of the chapter is Heredity and Evolution. And there are the few concepts which are deleted during this session 2020-21. So we will only focus on the important topics here. The first of all, as we know all that offsprings that we have discussed in the chapter 8, that offsprings are produced during reproduction, and offspring or the daughter cells, or you can say the child of the same parent, do not resemble with each other. When you see your brother and sister, then their faces, their features did not match with each other because there are some variations in the sexual reproduction. And we also see that many of characteristics of the children are same as their parents, grandparents and maternal grandparents also. And transmission of characters, traits from one generation to next generation is called heredity. And the branch of science in which we study heredity and variation is called genetics. So this is about heredity, and heredity and the variations are also there in the reproductive sexual reproduction. Now, who is the father of genetics? That is Mendel. Mendel chose pea plant or for his experiment due to following reasons. Very important. Sometimes this question is asked, why the Mendel selected pea plant for his study? Because availability of contrasting traits of several characters. Their lifespan is short. Normally self-pollination occurs, but cross-pollination can also be done by the artificial means, and large number of seeds are produced in the pea plant, which is very helpful for his study. Now, what are the rule of inheritance of traits? First of all, both the mother and father contribute practically equal amount of genetical material to the child. Second, chromosomes are located in the nucleus of each and every cell of human being. Third is chromosomes are made up of DNA and genes are the part of DNA. And genes are always found in the pairs in human beings and are called structural and functional units of heredity. This pair shows the contrast characters in which one is dominant, other is recessive. And this shows that each character can be influenced by both mother and father DNA equally. So, Mendel see the contrasting characters in the pea plant. There are so many character traits in the pea plant. One is dominant, another is recessive. If we see size of the plant, dominant is tall and recessive is short or dwarfed. If we see the shape of the seed, round and wrinkled. If we see the color of the seed, yellow and green. Color of flower, dominant is purple and recessive is white. So many contrasting characters we have taken by the Mendel to study pea plant. So one of the hybrid, there are two types of the cross are there. One is the monohybrid cross and dihybrid cross. In monohybrid cross, the cross between two plants with one pair of contrasting characters, for example tall and short, that is called monohybrid cross. And we can see first Mendel took two plants and they did the monohybrid cross. One plant is tall, other is short. If we see always gametes takes place in pair, tall and tall, short and short, you can see small t, then the gametes are capital T, its gametes are small t. So when they fertilize, then four pair of plants are there, four plants he obtained, capital T, if we cross multiply this, capital T, for example this is here, we can make a table, small t. If we multiply these, capital T, small t, capital T, small t, capital T, small t, capital T, small t. So in the first generation he found all the plants are tall and there is no difference in the size. First observation by the Mendel, that he found all the plants in the F1 generation were tall, no medium sized plants were there. But in the F2 progeny, when he took two plants, the same plants, he took, one is having both the characters capital T, small t, then he hybrid these plants. Then what kind of the gametes are there? Capital T, capital T and small t. And he took this table. Then male and female, that is the capital T small t, then he cross multiply capital T capital T, capital T small t, then small t capital. Always we have to put capital T before the small t, because dominant character always come first, and small t. Then in the F2 progeny, what he observed in the F2 progeny or in the second generation, 3 plants are tall. Only the one trait which is having the tall characteristics are sufficient to make the plant tall, and here is one is dwarf. So 3/4 are tall plants and 1/4 are dwarf. It means ratio is 3:1 in the second generation. So phenotype ratio means that appears phenotype 3:1, but in the genetically we see genes, one is TT and 2 are capital T small t, so 1:2:1. Capital T tall 1, capital T small t 2 are present, and 1 small t. So this is the genotypes. So what is the conclusion of Mendel experiment through this monohybrid cross? Capital T small t both are tall plants, and small t are short plants. First, single copy of capital T is enough to make the plant tall, while both the copies have to be T, then for the short plant. Characteristics or traits that like T is dominant trait because it expresses itself, and small t is called the recessive trait because it remains suppressed. Next is character that shows itself in the F1 generation is always a dominant character. That does not express itself is called recessive character or trait, and this is because of law of dominance given by Mendel. So this is about the monohybrid cross. Now this is about the dihybrid cross. In the dihybrid cross, dihybrid cross contain 2 plants having 2 pair of contrasting features. Just like here, we have seen then we took the 2 characters in the dihybrid cross, round and yellow seeds, dominant characters because they appear in the F1 generation. These are the conclusions. Then in the 2 yellow and green, so color is taken and the shape is also taken, round and wrinkled. So these 2 characteristics are taking 2 pair of contrasting characters are taking in this cross, so it is called dihybrid cross. And first the plant is yellow and red, yellow and round, and this is green and wrinkled. And after the gamete formations in the F1 generations, all the seeds are yellow and round. These are the dominant characters because they appeared in the F1 generations. Now the new phenotype combination shows that genes for the round, wrinkled or green yellow shades are inherent independently of each other. Then different 2 plants are taken, yellow and round, yellow and round. 2 plants were taken by them, and gametes are formed just like YYRR. So 2 gametes are there, you can see YYRR. If we 4 gametes are there, if we combine them, then you will get YY. These 4 are appeared in the first generation, capital Y, capital R, small y, capital R, capital Y. 4 gametes are formed after this fertilization. So male and a female, we put the female gametes from the heterozygotic parent or gametes from the heterogenic parent, male or female, both the plants, one plant and second plants, and if we cross multiply, then you will get phenotypes 9:3:3:1. And genetically we also can identify as we done in the upper example. So there are few questions, and on the basis of these 2 monohybrid and dihybrid, you can do these questions, and very important questions are there. I hope you understood it. And next one is sex determination. The factors responsible for the sex determination is that is, first of all, the temperature at which fertilized eggs are capped. They decide the gender. For example, turtle. Environment also is the one factor for determining the sex, that is also taking place in the turtle. And genetic, the gender is determined by the pair of chromosomes that are called sex chromosome. So sex determination is taking place on the basis of two factors. One is the environment, other is genetics. In the turtle, environment is responsible. In the genetics, the male and the female chromosomes are responsible, that are called sex chromosomes. So female XX sex chromosome and in the male XY. In human beings, there are 23 pair of chromosomes in male and the female and 22 pair of chromosomes similar in both male and female, but one chromosome is different. So one pair of chromosomes are different and 22 pair of chromosomes are called autosomes, which are similar in both male and female, but one pair of chromosome which is different in both male and female, which decide the gender, that are called sex chromosomes. And probability that the child born will be girl or boy is 50-50%, because XX or XY gametes are there in the male, XY in the female is X. So zygote formation is XX or XY, either XX or XY in the offsprings. If XX is there, then girl child is born. If XY is there, then a male or boy child is born. And all the children will inherit X chromosome from their mother, regardless whether they are boy or girl. Next is, thus the sex of the child is decided what they inherit from their father, not from the mother. So who decide the sex of the child? That is, who is responsible? Father is responsible. And next is, in human there are 50% probability of the birth of the boy and 50% probability of the girl child. So you can justify on the mechanism of sex determination in the human being. So you have to draw the diagram that is given above for justification and write these points. You can do these questions clearly. I hope you understood the concept and keep watching Super Vigyan. Thank you so much.